Hello, I'm Laura Marshall.
And I'm Melinda Rose, and this is Light Matters for August 14th, 2013.
On today's show, squeezed light is created on silicon, a laser inserts DNA into cells with a light touch, and invisible air turbulence is detected with lasers.
A microchip-based way to create squeezed light could assist a range of precision measurements and provide a viable route towards real-world on-chip sensor applications and technology. Monitoring a mechanical object's motion, even with a touch as gentle as that of light, fundamentally alters its dynamics. Squeezed light, with its quantum fluctuations below that of the vacuum field, was proposed nearly 3 decades ago as a way of overcoming the standard quantum limits in precision force measurements. In the past, squeezed light has been made using nonlinear materials with unusual optical properties. This latest work, engineered at Caltech, marks the first time that squeezed light has been produced using silicon, a standard material. The team engineered or punched holes into the silicon to make mechanical structures that respond to light in a very novel way that reduces noise. A detector measuring the noise in the light as a function of frequency showed that, in a range centered around 28 megahertz, the system produced light with less noise than what is present in a vacuum, which is the standard quantum limit. The team expects that with further improvements, the technology could be used to make integrated microscale devices for precision metrology applications. The work appears in Nature.
Gene therapy and genetic engineering always start with the same basic scientific question: how to transfect a cell by inserting foreign DNA into it. Many methods already exist, but they tend to be clumsy and destructive, not allowing researchers to precisely control how and when they insert the DNA, or requiring them to burn through large numbers of cells before they can get it into one. But a new method uses a femtosecond laser to poke a hole in the surface of a single cell and gently tug a piece of DNA through it with optical tweezers, offering greater control. Researchers in the School of Mechatronics at Gwangju Institute of Science and Technology in South Korea used the optical tweezers to grab hold of and transport a plasmid-coated particle to the surface of the cell. Guided by the trapped particle, they created a tiny pore in the cell membrane using an ultra-short laser pulse. While another laser beam detected the exact location of the cell membrane, they pushed the particle through the pore with the tweezers and dropped it into the cell, like a golfer sinking an easy putt. To determine if the method was successful, they inserted plasmids carrying a gene that codes for a green fluorescent protein. Once inside the cell, the gene became active and the cell's machinery began producing the protein. The green glow was then detected using a fluorescence microscope. They found that approximately 1 in 6 of the cells they studied became transfected. This rate is lower than that recorded for some other methods, but those are less precise and involve many cells at a time. What's special about the new method is that all this happens for one cell. The researchers hope the work will allow others to investigate the effects of transfection on individual cells, not just on large populations. The study was reported in Biomedical Optics Express.
A laser-based method of detecting unsteady airflow could provide a solution to sudden airplane turbulence. Clear air turbulence occurs independently of cloud cover, making it neither visible to the eye nor measurable with sensors. Recent results in atmospheric research indicate that such turbulence will occur more frequently in the future due to climate change. To address this, researchers at the German Aerospace Center's Institute of Atmospheric Physics developed a laser-based light detection and ranging, or LiDAR, instrument to be installed on board an aircraft. Through emitting shortwave UV laser radiation along the direction of flight, the density of the air is determined from backscatter values measured for the air molecules, oxygen, and nitrogen. Fluctuations in this density then provide information about turbulence in distant areas, making it visible and predictable for the first time. Under a European joint project, test flights are currently taking place on a modified Cessna jet operated by the National Aerospace Laboratory. The UV LiDAR system has been installed for the laser measurements.
Well, that's it for this edition of Light Matters, the Photonics Industry's only weekly newscast.
As always, you can write to us with your questions or comments at lightmatters@photonics.com.
Thanks for watching, and we'll see you next week.